Good morning. Myself, Dr. D.P. Pandey. I am going to share a class for you students. My topic is Introduction to the cell division and process of mitotic cell division. First, we should know that what is the cell division and why the cell used to divide. Cell division is a process by which a cell multiplies, or better to say, the old cells, they give rise to the new cells. And this concept was described first by the Fleming in 1882. It was first described by this gentleman, but its extensive studies were contributed by a gentleman which is known as Pandey in the year 1920. Means a cell used to multiply, or better to say, it used to increase its number by the process which is known as cell division. Why this occurs? We know that cell grows in size by the nutrients, and by these nutrients the nucleus and the cytoplasmic materials contained inside the cell, that increases, and a time comes when it needs its multiplication, otherwise it will destroy it. And this is the need of the cell division. Means its nucleus used to divide and produces another new cell. This is the cause of the cell division. We know that the organisms are of various types. The cells are of so many types. So, this cell division is of three types according to the nature of tissue or nature of cell. That three types of cell division, first is known as amitosis. Amitosis means A for not, and mitosis means equatorial division. means the division where the cell do not divide equally into the two daughter cells. That is known as mitosis. And this mitosis occurs in the embryonic membranes of vertebrates and in the different tissues like pathogenic and senescence tissues. This division says what? Here the nucleus divides unequally, means the chromosomal number which contained inside the nucleus that unequally divides into the two, and this type of division is known as what? Mitosis. The second division, it is the mitosis. Which we have today. Mitosis means the division in which a parent cell that divides into the two daughter cells. And the characteristic of this division is that in the daughter cells, the number of chromosomes that will be equal to its parent. Means, if it is a parent cell, it divides into the two daughter cells. Where the number of chromosomes, it will be equal to its parent cell. Means a diploid parent cell that divides and produces two diploid daughter cells, not haploid, two diploid daughter cells. These are the daughter cells. And it is the parent cell. The parent cell divides into the two diploid daughter cells, and this type of division is known as mitosis. And all the growth means from the zygote up to the adult stage. This mitosis is responsible. and it always occurs into the body cells. It occurs into the body cells or better to say into the somatic cells. Means the growth of the body, a seedling that is going to be planted and it is growing, it is increasing in size, and ultimately it is forming a plant, big tree. Likewise, a young born baby, it increases in its size, means it grows up to the adult stage. And for this increasement in the size, this increasement in the organ, that is done due to the process that is known as what? Mitosis, where cell divides and divides and divides repeatedly. So this mitosis is also known as equatorial division. This is the equatorial division. Now, the third division, it is known as meiosis. It is also known as reductional division. Here, in the meiosis, the cell divides twice. And it always occurs into the germ cells. Or better to say into the gonads. Inside the body of any organism, there exist two types of cells. The first cell, that is known as somatic cell, and the another that is known as germ cell. As I described that into the somatic cells, mitosis always occurs, but there are exceptions because this biology is the science of exception. There no theory exists 100%. It contains always an exception. There are some somatic cells where sometimes meiosis always takes place, but permanently this meiosis occurs into the germ cells. And as I described, what? That here the cell divides twice. If it is a parent cell having diploid number of chromosome. First, it divides into the two, having n number of chromosome. Here a diploid parent cell, it is giving rise to daughter cells where the chromosomal number is going to reduce just half. And so this first division is known as first meiotic division, or better to say reductional division. Here, the reduction of chromosomes takes place. And its second division, here, it is just like mitosis. It is just like mitosis. Means, here, this single cell, it will produce two daughter cells. Here also, it is going to produce two daughter cells. What is occurring here? That this haploid number of daughter cell, it is giving rise to haploid number of daughter cells. And so, it is known as second meiotic division. Or mitotic division. What is happening here? A single parent cell, here it is a parent cell. It is giving rise to four daughter cells, 1, 2, 3 and 4. These are the daughter cells. And into the germ cells, always this type of division takes place. And as we know that the gametes produced by the gametes produced by the gonads, these having this chromosomal number, haploid chromosomal number in these daughter cells, there will be development. It will develop into the either into the sperm. If it is male gonad, it will produce the sperm. If it is female gonad, it will produce ova, and it is required into the sexual reproduction. If this meiosis doesn't occur, the sexual reproduction, it will not take place. And if it will take place, then the constant chromosomal number does not exist. For the constant chromosomal number, this meiosis is badly required. Now we have discussed what is cell division and the types of cell division. Now I am trying to explain the process of mitotic cell division. This mitotic cell division, as we have discussed, that here it always occurs into the somatic cells. And what is the process? That a diploid parent cell, it will produce two diploid daughter cells. How it happens? If I have to distribute, suppose I have 2 rupees, I have to distribute it among 2 students. And both are getting 2 rupee and 2 rupee each. How it is possible? Means there exists certain process by which this 2 rupee, it is going to multiply by 2, means 2 into 2 is equal to 4. Then I can distribute it among 2 students equally. And this doesn't occur in the meiosis. So, this mitosis cell division processes. These 2 processes are involved into the mitotic cell division, mitosis. The first, it is the interphase, where cell in this phase, into the interphase, the cell doesn't divide. Better to say, it is the preparatory phase before division. And this mitotic phase is the phase of division. Means this interphase, during this interphase, the cell prepares itself for the division. The required materials, the required DNA, means the duplication of DNA. What I told, that here is the need to duplicate the chromosomal number, to increase the chromosomal number. And this increasement As we know that the chromosomes are formed by the DNA. The genes are formed by the DNA. And for the duplication of chromosome, the DNA synthesis is very much essential. If the increasement of DNA doesn't occur, then it cannot be divided equally, means equal chromosomal number will not exist there. And keeping the view in our mind, if we are going to consider this interphase and mitotic phase, and what is the interphase and what occurs during the interphase and what occurs during the mitotic phase, then if we are going to draw a cycle of the interphase and mitotic phase, then the major time, major hours, it is contained into the interphase. Like from here to here, this is the interface from this place up to this place. Here, this interface has been divided into the three subphases. The first subphase, it is known as G1. Growth phase 1. First, we should know that this interphase is also known as resting phase. As I discussed, that during this interphase, no division will take place and cell prepares itself for its division. We are into the G1 phase. This is the purely preparatory phase. The proteins and the other essential materials required for the synthesis of DNA, this is going to be stored by the cells during this G1 phase. And this is the longest phase. Means the maximum time that is required into the phase. If this cycle is of a mouse, अगर एक माउस का चूहे के अंदर where a cell going to divide means where a cell is doubling in 24 hours. Here, this G1 phase, it will take 14 to 16 hours of time. Then, all the material required for the synthesis of DNA, it is going to be accumulated into this G1 phase. Then now, the cell is ready to synthesize its DNA because all the material required for the DNA that has been stored during this phase. So this second phase, this is the S phase. Better to say synthesis phase. Here the cell prepares, or you can say synthesizes the DNA during this S phase. And the time, it will be 6 to 8 hours. It will need 6 to 8 hours of time. One thing which I want to share with you, that sometimes, not always, sometimes as this G1 phase starts, it may be possible that all its function, it can ceases out. It may be ceased out here, it may be ceased out here, it may be ceased out here. Suppose here it is going to cease. Then further it will not develop. It is not going to enter into the S phase. Here again, it will start from this place. Means G1 phase again going to be started. As soon as it is discontinued at any place, if ceasing of the process at that place, from that place, it will again re-enter into the given phase, and this phase is known as G0 phase. G0 phase means G0 phase will indicate that all the processes going to be ceased during the given phase, and it is again going to enter into the given phase. And as soon as this given phase ends, this S phase, it will be started, where the duplication of DNA, the synthesis of DNA, it takes place. And this third phase of the interphase, it is known as G2 phase. During this G2 phase, the cell inspects itself before its division, and if required, if anything is required except DNA synthesis, it will be stored or it will be occurred at that G2 phase. Cell inspects before its division. अभी बरसात का समय है, इस समय गांव वगैरह में बरसात के समय में गार्जियंस बोलते हैं, व्हाट दे टेल्स, जो गार्जियन है वो कहते हैं कि भाई देखो, there is no gas facility. I am talking about that place. वहां कहते हैं कि भाई देख लो, 2-3 महीने जो है वो जलावन नहीं मिलेगा, इतना स्टोर्ड है, देख लो, और उससे काम चलाना है. This is the inspection. Here also, the cell inspects itself. कि अगर किसी चीज की कमी है, then I have to prepare for that. And as soon as this ends, means measure of the division, here it will take 4 to 6 hours of time. G2 phase. And after this G2, the phase which is the phase of division. This is the M phase. This is the M phase. And this M phase means it is the phase of division. Here the cell starts to divide. And I am talking about the mitosis into the mouse. चूहे की बात यहाँ पर, where in 24 hours the cell divides and produces double number of cells. Hence single mitosis. Here in 1 and 2 hours, a number of processes are involved, and means a number of changes occurs during this M phase, during this division phase, and so we have divided it into a number of states. The first state, it is known as prophase. The second state, it is known as metaphase. The third state, it is known as interphase. The fourth, it is the telophase. This is the phase of division. Hence, this is the mitotic process, and ultimately, the last process, which is known as cytokinesis. Cytokinesis means the division of cytoplasm. And after this cytokinesis, the two daughter cells, it will be produced, which will contain the diploid number of protein, which will be the equal of its parent, so it is the equatorial division. Am I clear? Now, we will proceed for this M phase. What are the changes which takes place during this process? This we have to discuss. First, prophase. This is the interphase. Interphase nucleus. Here the cell attains its full size due to the accumulation of the cytoplasmic matrix. It is the chromatin network. This is the nucleolus. This is the chromatin. This is the centriole. What happens here? It is the early prophase. What happens here? That this first the centriole used to divide or used to split up into the two sets, and they move towards their opposite poles. Here the chromatin network, here, the chromatin networks, it becomes short and thick, and this is going from the chromosome which contain centromere. Here is the nucleolus. This is the early prophase. The nucleolus is going to slowly, it is disappearing. Here the nuclear membrane, it is also disappearing. This is the middle prophase. And the centriole, which are moving to their opposite poles, around them there are certain rays-like structure which appears, and this is known as asters or astral rays. This is known as aster or astral rays, and these are formed by the microtubules present inside the cytoplasm. which is the late interphase, where the nucleolus being disappeared, the nuclear membrane, it will be vanished, and the chromosomes which contains the centromere, they will be inside this nucleus, and from this centriole there emerges certain race-like structure formed by the microtubules, and these structures are known as spindle fibers. And as soon as the nuclear membrane and nucleolus these disappears, it indicates the end of the phase. And sorry, my other colleagues are ready to take their classes on their time. So I require more time to explain these, and so I will give some another time to you. Stay with me.